Hello, everyone. This is my Foundations of Evolution presentation. So my topics are pollination biology, community ecology, and spatial ecology. Pollination biology, we will be going over the pre-experimental pollination from 1761 to 1925, the experimental pollination, which is a book that kind of changed the field, evolutionary pollination, from 1949 to 1970, just trying to understand the relationship between pollinators and the flowers and how they drive each other. The pollination revolution from 1983 to 1999, and interpreting pollination networks. So let's begin. The pre-experimental pollination research was mostly just observing pollinators in their natural habitats, just kind of taking a look and trying to look at patterns. And so Springle in 1793 was looking at flower symmetry and how that kind of impacts pollinators and how it drives them to visit, as well as orientation, which he didn't believe impacted pollinators, and nectar guides, which he was the first person to really understand that the purple that you see on the inside of those flowers is actually there to help guide the pollinator in to get the nectar. And so understanding those mechanisms. Then you have Darwin in 1876, who was analyzing the effect of fertilization. And so he wanted to understand how cross-fertilization versus self-fertilization impacted the progeny of the plant. And so what he ended up finding was that some plants will self-fertilize more readily than others, and that the ones that do tend to have different floral syndromes than those that don't, and that the ones that don't rely on self-fertilization tend to have more attractive flowers to pollinators. He was also one of the first people to observe or document nectar robbing, which you can see this bee in the corner doing, which is just instead of pollinating, going in and nibbling a hole into the flower and drinking its nectar. Then we have experimental pollination in 1925, which was written by Clements and Long. And in this book, there are about 100 experiments that they do that they criticize previous pollination biologists or people who are looking at pollinators and flowers for not doing experimentation. And so what they did was they took flowers and changed the position and saw if that impacted pollinator visitation. They concealed and disguised flowers. They painted them. They had artificial flowers. They mutilated them, removing petals or even adding petals. They had different flowers next to each other to understand competition between pollinators. And they manipulated the nectar of them as well by adding things like honey or vinegar to the nectar to see if that maybe enticed the pollinators to visit more or less. And so here are some examples of the orientation. For instance, you have the upside down flower versus the right side up. You have the two different bees visiting two different fake flowers, one with nectar, one without nectar. And then you have the flowers that have removed petals, they are mutilated, versus the one that doesn't. And so from there we move on to evolutionary pollination with Vern Grant, who examined the taxonomic characteristics of flowers in different regions and noticed that different regions have different pollinators as well as different flowers, and that the pollinators confine their visits to certain kinds of flowers. So you're going to have specialization is the idea, more so specialization than generalization, which is kind of contrary to what the thought process is today. Further, he posited that pollinators can actually drive evolution through ethological evolution, so behavioral. And so say you have a pollinator that is visiting one species of poppy, and that species has two different phenological types, say one's purple and one's orange, and this pollinator is only pollinating the purple ones, then it's selecting for that purple trait. And so it could drive evolution. Then you had Stebbins in 1970, who kind of took it a step further and really looked into pollinator syndromes. So at this point, there was disagreement about whether or not floral diversity that everyone saw in angiosperms was driven by pollinators, like we know it is today. And so what he examined was the adaptive radiation in orchids and how it was driven by pollinator vectors or animal vectors, and how these radiations are influenced by the most effective pollinator. So if you have 10 pollinators pollinating a plant, but one pollinates the best, the one that pollinates the best is going to lead or is going to have more evolutionary pressure than other ones. And then you can see the two examples of syndromes that I have up here with the orchid wasp, which thinks that it's currently mating with a female when it isn't, it's actually pollinating, or this sicklebill hummingbird whose beak co-evolved with this flower to get that nectar perfectly curved. From there, we move into what I call the pollination revolution, which happens from the early 80s through the 1990s. And during this time period, there is just a boom of pollinator papers. There's tons of research going on. And it kind of starts with the Handbook of Experimental Pollination, which goes further in depth from where Clements and Long left off, by trying to create the best ways to ask pollinator ecology questions, by going through a series of other ways of devising these questions. So they went through that. They wanted to also create a uniform way to ask these questions. And so that's kind of what this book did, in addition to also having experimental methodologies. Then we move further on to Giordano, 1987, who was the first to collect data that could be used in a network format. He didn't use it in the network format. It would be Mehmet in 1999 who does for the first time, but he's one of those precursors. And so he also noticed that more species leads to more interactions within communities for pollination. And he also realized that connectance decreases while species richness increases, but differently in different pollinators. So for instance, in smaller pollinators, like the euglossin bees right there, you're going to see a lower overall rate of connectance compared to hummingbirds and bumblebees, which are large and can travel between these different communities, whereas you're not going to see that with small bees. They're not going to travel as far, for instance. Then you have Mehmet in 1999, who creates the first plant pollinator network, which is impressive because this is still used today. It's still a very common tool for understanding the interactions between the plants and the pollinators. And so each one of these little black bars is a pollinator species at the top, and at the bottom, they're plant species. And so each of those species, the wider it is, the more individuals there are, the more it was sampled. The narrower, the less it is. And so, yeah, you can kind of see how this will show you a relationship in an ecosystem. From there, we go into interpreting pollination networks, which is 2007 to present, from Olsen et al., who, rather than looking at it in that format that we previously saw with the lines connecting everything it's more of a presence absence in a grid format, which is also used today. Because by doing this, you can actually manipulate your data to run null models and things like that and see if your distributions are random or if there's actual meaning to them. And you can see in the B graph that these relationships are modular. So that means that that they're kind of confounded, that you're not seeing too much generalization. It's a lot of specialization. And that's what each one of these little modules kind of represents. And so he was one of the first people to discuss modularity and nestedness. And that's why this paper is so impressive. So now we move on to community ecology, where we're gonna go through everything. So you have the general morphologies of organisms from 1866 to 1890, which is created by Ernst Haeckel, who is a morphologist. He studies morphologies in organisms, but he's actually the founder of ecology as a field. And so he sought to create a general doctrine of morphologies to understand how natural selection and common descent can be integrated into morphology. And so he worked closely with Darwin to create these ideas, but he classified ecology as the physiology of relationships and later expanded upon it to be the relationships between organisms and their environment and the conditions of existence. But he was not without flaw. He didn't really understand quantitative ecology and actual experimentation. And so Henson in 1890 did several plankton studies where he sampled a ton of plankton communities and used his data and some statistics to predict where you're going to find the most plankton abundance and the least plankton abundance. And so by doing this, he predicted that in warmer waters, you will find fewer plankton than you will in colder waters. And so when he did this, Haeckel objected because he was using data that he didn't have. He was predicting something, and Haeckel kind of thought that statistics was a hand-wavy science, that it wasn't real. And so I think we all know who ended up being correct in this one. But yeah, not without flaw. And so then you have The Lake as a Microcosm by S.A. Forbes, which I think is an extremely important ecology paper for US ecologists, because it was one of the first surveys where Forbes went through and records the plants, algae, diatoms, invertebrates, fish, everything in these lakes, and compares them historically to one another, and notices that there's a pattern, that there's a trophic pattern specifically, that you're getting a ton of invertebrates, which get eaten by a ton of small fish, which get eaten by fewer medium fish, and then fewer large fish on top of that. So you kind of have this cascading effect. And so he was one of the first people to kind of come up with the idea of a food web by noticing this. And that also included applying these things to natural selection. And so understanding that in a lake, these predators aren't going to be able to move around all that easily between lakes and things like that. And so the most efficient predator is going to win out relatively quickly because they can't escape. Further, You have Clements in 1916, who comes up with the term biome when he's trying to categorize plant communities. And additionally, he was trying to understand how succession works between these communities and was defining these different communities as biomes. They're different biomes was the idea. And from there, you get Tansley in 1935, who expands on it. He thinks that biomes are necessary, but they're insufficient because you're not getting the full picture. You're not getting the abiotic factors in addition to these biotic factors. And so he creates the new term ecosystem, which not only includes organisms, but the physical conditions in which they reside. And then after that, you end up getting into niche and theoretical ecology, with Savage in 1957 defining the term ecological niche in response to Ross. So the term niche existed before this, but it was often misused and synonymous with habitat. It wasn't until Savage in 1957 really laid down the law, telling Ross that organisms that are similar and are in similar habitats occupy different niches when they are faced with different predators and different parasites and things of that nature. And that those interactions are part of what a niche is, not just its habitat. Then you get Hutchinson in 1959, who theorizes why there are so many kinds of animals. And he kind of thinks about it and concludes that smaller groups of organisms aren't going to have as much genetic, aren't going to have as large of a gene pool as larger groups of organisms. So when you have larger groups of organisms, you're going to see more gene diversity, or you're going to see more gene diversity, and thus more evolution. And then he carries it on to also be that smaller organisms are going to see more diversity because they can occupy more niches and diversify into these. So thinking of bugs versus elephants, for instance, there are way more beetles than there are elephant species, because there are so many niches for them to occupy. And they also just have much larger gene pools. So then from there, we kind of go to the unified neutral theory of biodiversity, which was proposed by Hubble in 2001. And he proposes this to say that all organisms are functionally equivalent and that biodiversity is controlled by neutral drift rather than niche partitioning. And an example that he uses of this is phenotypic plasticity, because if organisms can show different or occupy different niches in these different environments, then they're functionally equivalent to each other. So a plant that can be both a tree and a shrub can be equal to either a tree or a shrub, for instance. And so that was derided pretty quickly because there are tons of examples of niches leading to biodiversity, for instance, with coral reefs. There are so many different niches in a coral reef community that you end up having so much diversity. And so this idea was not received well. And then Zhu Zhang in 2008 proposed the nearly neutral theory, which rather than saying that these organisms are all equivalent, that they're close to being equivalent, that they do have different fitnesses, but by occupying different niches, they're able to kind of they're able to equalize these niches. And so they're nearly neutral. And as a result, that's why an organism with a lower fecundity rate is not going to be driven to extinction. From there, we get into spatial ecology. And so we're going to start off with patterns in spaces and species interaction. And so Watt in 1947 was the first ecologist to observe spatial components within plant communities. And so he was looking at the reasons why plants occur in patches versus areas without plants and things of that nature. And he concluded that drought was part of the reason for that. He also posited that the reason why when you look at certain plants, certain areas, you're going to see certain patterns, much like this photo right here, is because these organisms are close enough to each other to not have to compete much for resources. But if a seedling tries to grow between them, for instance, then it's going to have to compete with both of those plants. And that's why you won't see seedlings or plants between these. And so it creates this pattern of how close can these plants get without having to interact with each other. And then Huffaker in 1958 examines the role of dispersion in predator-prey cycles, which is super neat. He used predatory and prey mites and had oranges be the amount of space they could occupy. So he released the same amount of mites, 25 mites, or 25 prey mites and 2 predator mites onto either 10, 20, or 40 oranges. And so he would release them onto the 10 oranges, the prey would disperse, as you can see in the top graph, and their population would increase. Then the predators would increase as well and drive the prey to extinction, and both would go to extinction. But when these prey mites had 40 oranges to expand upon instead, so they could avoid predation, they were able to survive for 8 months rather than 2 months. And so you end up seeing this cycle of the populations going up and then down and up and then down as the predator population goes up and then down. And so then we move on to the distribution of organisms with Skellum 1951, who derived the ecological diffusion model from molecular models and then used them to understand how space can alter populations and community dynamics. He then compared this to muskrat data that he had, which you can see in the bottom left, how it in 1905 is in a certain spot, and by 1927 it's expanded. He looked at the area of the expansion and compared his models to it, and his models lined up with that data. And as a result, these models are still used today for looking into things like invasion. And then from there, we move to Kariva in 1983, where we take that idea of invasion and we kind of look at it on the individual level of these organisms. And so we're going to see how do they move. And so Kariva wanted to understand the movements for ovipositing butterflies and for foraging butterflies and for their larvae, the caterpillars. And so you can kind of see in the A and B the idea of how Kariva drives his equation here. It's the distance between all of these spaces. And so for A and B, A is the random walk prediction in the non-dark circles, and the dark circles are the actual data that they had for when these butterflies were looking for locations to oviposit. And so they were able to predict how many moves and how far these butterflies were willing to go to oviposit and during ovipositing, which is impressive. However, it did not work for foraging because their explanation was that foraging is non-random. These butterflies aren't looking for a random spot to forage, they're looking for a spot. Then they tried to apply it to the larvae and found that that worked not so well, that it was iffy because the caterpillars would move in non-random fashion as well. They would go left and right, left and right, left and right in a non-random way. And so that model didn't work for some of these, but it surprisingly did work for oviposition. Now we move on to conservation practices in spatial ecology. Higgs and Usher in 1980 used spatial formulas to discuss whether or not nature preserves should be large or small, whether or not you should have 2 large preserves or 6 small preserves of the equal or of the same amount of area. And so using some equations and deriving them, they were able to create an equation to predict how much diversity you would get out of a large preserve versus a small preserve. And it comes down to the P versus Z value. So for instance, if you have a P value of 0.4, your Z has to be about 0.3 in order to have it justify using multiple sites rather than using one large site. And so he compared this to actual quarry reserves and found that some of them, found that all of them would work better as multiple reserves rather than the large reserves that they were, but mostly that some of them would work better than others as well. So there's that. And finally, we get to the biodiversity hotspot thesis, which is the idea that certain areas are hotspots for biodiversity, that you have a ton of organisms in the Amazon, in an acre of the Amazon rainforest versus an acre of Nebraska, that it makes more sense to preserve that 1 acre in the Amazon rainforest because you're going to get more bang for your buck. And so this idea was proposed by Meyer to enact conservation efforts and ended up being able to raise $1 billion to preserve hotspots as they defined them, which ended up being just extremely dense biodiversity areas. And so these hotspots, after they were able to, these hotspots ended up being criticized because by drawing resources away from other conservation efforts that are less biodiverse, they ended up harming these other ecological services. So even if Nebraska doesn't have as much biodiversity, it still has ecological services that should be preserved. And so that was one of the arguments for keeping these cold spots, to which the other, the hotspot advocates would argue, well, we're not saying that we should defund conservation in Nebraska per se. We're saying that we should just fund more towards these hotspot areas. So that's kind of where that argument ended up going. There's still people discussing it on both sides, but it mostly looks like the hotspot people have won out. And then here's a map of all of the biodiversity hotspots, so you can examine it. And it's mostly been adopted by all of the conservation organizations and NGOs and things like that. All right, thank you very much. Goodbye.